Before examining bacterial structures individually, it is essential to establish the scientific context. In the field of microbiology, the bacterial cell represents a unit of life that is relatively simple in structure compared to eukaryotes, yet remarkably complex in function. Although bacteria lack a true nucleus, they possess various, coordinated structures designed to maintain viability and adapt to diverse environments. The invention of the first prototype electron microscope in 1931 by Ernst Ruska and Max Knoll, followed by its commercial introduction in the 1940s, marked a pivotal turning point in science. This technology provided the high resolving power necessary for the accurate observation of the cell's fine structure. By utilizing electron beams instead of light, electron microscopy achieves superior resolution, even allowing for the visualization of atomic structures in modern applications. Technically, the standard unit of measurement for bacteria is the micrometer, which is equivalent to 1,000th of a millimeters, 10 to the power of -3 millimeters. The dimensions of bacterial cells are frequently determined using an ocular micrometer, a graduated disc inserted into the microscope's eyepiece. To ensure precise measurements, this instrument must be calibrated against a stage micrometer. The following sections systematically analyze the structural components of bacteria, proceeding from the outermost layers to the internal regions. Flagella. The first component to consider is the flagellum, plural, flagella, a thin, whip-like appendage that protrudes through the cell wall. Originating from a basal body embedded within the cell, flagella primarily serve as organs of locomotion. Flagella are not universal to all bacteria. They are commonly found in bacilli and spirilla but are rare in cocci. Structurally, a flagellum consists of three primary parts. 1. The basal body, which functions as a motor embedded in the cell envelope. 2. The hook, a connector between the basal body and the filament. And 3. The long external filament. The length of a flagellum can be several times that of the cell itself, though its diameter is extremely minute, ranging from 10 to 20 nanometer. The structure is composed of protein subunits known as flagellin. Due to their narrow diameter, flagella cannot be visualized under a standard light microscope without specialized treatment. Through staining procedures involving a mordant, a substance that precipitates dye onto the flagella, the diameter can be artificially increased until the structures become visible. Functionally, flagella are responsible for motility, enabling bacteria to perform chemotaxis, which is the directed movement in response to chemical stimuli. This capability is crucial for pathogenic bacteria to reach and invade host tissues. Furthermore, flagella are highly antigenic and can be recognized by the host's immune system. Pili and fimbriae. Next are the pili and fimbriae, which are thin, filamentous appendages protruding from the cell surface, predominantly found in gram-negative bacteria. Morphologically, these structures are smaller, shorter, and more numerous than flagella, necessitating the use of electron microscopy for visualization. Although these terms are often used interchangeably, there is a generally accepted functional distinction. Fimbriae occur in large numbers and function primarily in adhesion to host cell surfaces or substrates, whereas pili or sex pili are usually limited to 1 or 2 per cell and facilitate conjugation, the transfer of genetic material between bacteria. It should be noted that while they are generally not used for swimming, type 4 pili enable a specific form of movement known as twitching motility on solid surfaces. Because of their role in adherence, fimbriae and pili are significant virulence factors. Antibodies targeting these structures can inhibit infection, making them prime targets for vaccine development. Capsule and slime layer. Extending beyond the cell wall, many bacteria possess an additional layer known as a capsule. Capsule and slime layer. Extending beyond the cell wall, many bacteria possess an additional layer known as a capsule. While not present in all species, the capsule is vital for pathogens such as Streptococcus pneumoniae. These capsules are typically composed of polysaccharides, though certain species, such as Bacillus anthracis, produce capsules made of polypeptides. The thickness of the capsule is often influenced by the growth medium. Capsular material is secreted by the cell and, due to its high viscosity, remains firmly attached to the cell wall. Encapsulated bacteria grown in liquid media may cause the medium to become ropey due to the accumulation of this material. The functions of the capsule include protection against desiccation and harsh environmental conditions, acting as a nutrient reserve, and inhibiting phagocytosis by the host's immune system, thereby enhancing virulence. Additionally, capsules facilitate adhesion and biofilm formation. If a bacterium loses its capsule, for instance, through mutation, it often suffers a significant loss in virulence. A distinction must be made between a capsule and a slime layer. A capsule is an organized, viscous structure firmly attached to the cell wall, whereas a slime layer is more fluid, unorganized, and easily detached or diffused into the surrounding medium. Special accessory structures, sheaths and stalks. Certain bacterial species possess specialized structures that are not universally present across all taxa. These are typically adaptations to specific environmental niches. Sheaths are predominantly found in specific bacteria inhabiting freshwater and marine environments, such as iron bacteria. A sheath is a hollow tubule or cylindrical envelope that encloses a chain of cells. In many instances, these sheaths are reinforced by the deposition of insoluble metal compounds, such as iron or manganese oxides, which arise as byproducts of cellular metabolism. The primary functions of the sheath include providing cellular protection, maintaining structural morphology, and facilitating attachment to substrates. Stalks are exhibited by certain species, such as Colobacter, which develop semi-rigid appendages that extend from the cell body. The distal end of the stalk contains an adhesive substance, often referred to as a holdfast, which enables the bacterium to adhere firmly to solid surfaces. This mechanism is vital for bacteria residing in high-velocity aquatic environments, as it prevents them from being swept away by the current and ensures they remain positioned in nutrient-rich zones. The cell wall. The cell wall constitutes a primary, structural layer located beneath the capsule or slime layer, and external to the cytoplasmic membrane. Its functions are vital to cellular viability. It defines the cell's shape, provides mechanical rigidity, and protects the cell from osmotic pressure, thereby preventing osmotic lysis in hypotonic environments. The cell wall typically accounts for 10% to 40% of the bacterial dry weight. The fundamental component of the cell wall is peptidoglycan, murine, a heteropolymer composed of N-acetylglucosamine (NAG) and N-acetylmuramic acid (NAM), linked by peptide chains. This intricate structure is the primary pharmacological target for beta-lactam antibiotics, such as penicillin and cephalosporins, which function by inhibiting the cross-linking of peptidoglycan strands. Based on the Gram-Stain reaction, bacterial cell walls are categorized into two distinct types. 1. Gram-positive bacteria. These are characterized by a thick peptidoglycan layer, 15 to 80 nanometers, and the presence of teichoic acids embedded within the matrix. They lack an outer membrane. While generally more susceptible to antibiotics targeting peptidoglycan synthesis due to the accessibility of the thick layer, they exhibit greater resistance to certain physical-mechanical disruptions compared to gram-negative organisms. 2. Gram-negative bacteria. These possess a significantly thinner peptidoglycan layer, which is shielded by an outer membrane, containing lipopolysaccharides, LPS, porins, and lipoproteins. This outer membrane renders gram-negative bacteria less susceptible to penicillin due to permeability barriers and more resistant to various chemical agents. However, they are more vulnerable to osmotic disruption if the outer membrane is compromised. Notably, LPS acts as an endotoxin. Upon bacterial lysis, it can elicit a severe immune response leading to septic shock. Cytoplasmic membrane. Situated immediately beneath the cell wall is the cytoplasmic membrane or cell membrane, measuring approximately 7.5 nanometers in thickness. This membrane serves as the critical boundary separating the cytoplasm from the external environment. Its architecture conforms to the fluid mosaic model, consisting of a phospholipid bilayer with hydrophilic heads oriented outward and hydrophobic tails sequestered in the center, interspersed with various integral and peripheral proteins. The primary functions of the cytoplasmic membrane include selective permeability, regulating the molecular traffic entering and exiting the cell. Passive diffusion, the movement of substances from high to low concentration without energy expenditure. Active transport, the movement of solutes against a concentration gradient, requiring metabolic energy and specific carrier proteins, a process essential for concentrating nutrients in oligotrophic environments. Metabolism, serving as the site for enzymatic reactions, such as the electron transport chain in aerobic bacteria. Environmental sensing, containing receptors that detect chemical and physical fluctuations in the surroundings. The integrity of this membrane can be compromised by antibiotics such as polymyxins, which induce leakage of cellular contents. Note on mesosomes. In historical literature, the cytoplasmic membrane was described as invaginating to form mesosomes, which were thought to assist in cell division. However, modern research utilizing cryogenic electron microscopy, avoiding harsh chemical fixation, has demonstrated that mesosomes are preparation artifacts and do not exist in living bacterial cells. Consequently, the biological function of mesosomes is no longer recognized in contemporary microbiology. Cytoplasm and internal components. The cytoplasm is a gelatinous matrix that fills the interior of the membrane, serving as the primary site for cellular metabolism, including glycolysis and biosynthesis. It houses the following components: ribosomes. These are RNA protein complexes responsible for protein synthesis, translation. Bacterial ribosomes are characterized as 70S units. The S refers to Svedberg units, which denote the sedimentation rate during ultracentrifugation. They consist of a small 30S subunit and a large 50S subunit. The structural divergence between these and eukaryotic 80S ribosomes allows for selective toxicity by antibiotics such as tetracycline and chloramphenicol. Nucleoid, an irregularly shaped region within the cytoplasm, containing the genetic material. Because bacteria lack a nuclear membrane, the DNA, typically a single, circular, double-stranded chromosome, is condensed by DNA-binding proteins within the nucleoid. Plasmids, small, extrachromosomal circular DNA molecules capable of autonomous replication. Plasmids often carry non-essential but advantageous genes, such as resistance factors, R factors, which confer antibiotic resistance, virulence factors, determining microbial pathogenicity, and specialized metabolic genes. Plasmids can be transferred between bacteria via conjugation, driving evolution and the spread of drug resistance. Inclusion bodies, cytoplasmic granules that function as nutrient reserves, e.g., polyphosphate, glycogen, or sulfur, or specialized structures like chromatophores in photosynthetic bacteria. Protoplasts and spheroplasts. Understanding the removal of the cell wall is essential for elucidating its protective role. When the cell wall is enzymatically degraded, e.g., by lysozyme, in an isotonic solution, two forms may emerge: protoplasts, formed from gram-positive bacteria. These cells lose the cell wall entirely, becoming spherical and highly sensitive to osmotic changes. They generally lose the ability to divide. Spheroplasts, formed from gram-negative bacteria, where the presence of the outer membrane results in the retention of some wall components even after peptidoglycan removal. Both forms are prone to osmotic lysis if transferred to a hypotonic solution. Bacterial spores. To survive deleterious environmental conditions, some bacteria undergo sporulation. Exospores, formed outside the vegetative cell, such as the conidia of Streptomyces, which resemble fungal spores. Endospores, developed within the vegetative cell. These structures are exceptionally resistant to heat, radiation, and chemical disinfectants. Endospores are produced by certain species within the genera Bacillus, Clostridium, and Sporus arsina. Key characteristics of endospores include the presence of dipicolinic acid 5 to 10% of dry weight, complexed with calcium, which confers thermal resistance, and a thick protective layering comprising the cortex and spore coat. In this state, metabolism is dormant. The position of the spore central, terminal, or subterminal and its size relative to the cell are diagnostic features in bacterial identification. Upon the return of favorable conditions, the spore undergoes germination, progressing through activation, germination, and outgrowth, to return to an active vegetative state. The bacterial cell is a highly organized biological system. Every component, from flagella for motility and capsules, for protection to the cell wall for structural integrity, and the nucleoid for genetic information, operates synergistically. A comprehensive understanding of these structures is not only fundamental to basic biology, but is also critical for the advancement of antibiotics, vaccines, and effective infection control strategies.